Hello and welcome to my channel. In today's video, we are going to discuss about nucleic acids. Nucleic acids, they are biopolymers consisting of long chain of monomers. And these monomers are called nucleotides. Biopolymers means biological polymers, which means they are biologically derived and not synthetically derived. And they are polymers, which means they are made up of multiple monomers. The monomers of nucleic acids are called nucleotides. Nucleic acids are of two types, DNA, that is deoxyribonucleic acid, and RNA, ribonucleic acid. The main functions of nucleic acids involve they store genetic information for cellular growth and reproduction. Now let's understand what is a polymer before we continue. So polymers, majorly the biological polymers are of four types: carbohydrates, lipids, nucleic acids, and proteins. All of them are made up of multiple monomers. For example, proteins are made up of amino acids. Carbohydrates are made up of monosaccharides. Similarly, nucleic acids are made up of nucleotides. Now let's see the nucleotide structure. A nucleotide is made up of three components: a sugar, phosphate molecule, and a nitrogenous base. First of all, sugar. This is a 5-carbon sugar molecule, carbohydrate. Just like you must have heard about 6-carbon sugars, for example, glucose, they're called hexoses. Similarly, the 5-carbon sugar, 5-carbon monosaccharides, they are called pentoses. The most important pentoses which we'll be learning in nucleotides are of two types: ribose and deoxyribose. Deoxyribose sugar is found in DNA and ribose sugar is found in RNA. 5-carbon sugar molecule has 5 carbon in its structure, starting from carbon number 1, 2, 3, 4, and 5th carbon is outside. The difference between ribose and deoxyribose sugar is at the second carbon, ribose sugar has HOH group, whereas deoxyribose has no OH, just H. Second component is nitrogenous base. This is called nitrogenous because it has nitrogen along with carbon, hydrogen, oxygen, which is present in almost all the macromolecules, biological polymers. They're called bases because they donate electrons to other molecules. Nitrogenous bases are of two types: purines with 9-membered ring and pyrimidine with 6-membered ring. There are two types of purines, adenine and guanine, and there are 3 pyrimidines, cytosine and thymine in DNA, cytosine and uracil in RNA. So DNA has thymine, whereas RNA has its alternative version called uracil. The third component of the nucleotide, that is the phosphate group, if you have seen the structure of ATP, the structure looks just similar to ATP, adenosine triphosphate. If you add to this adenine group a ribose sugar, it will become adenosine. And then add a phosphate group, it becomes adenosine triphosphate because of the 3 phosphate groups. Phosphate group oxygen, which you can see here, is a polar molecule and has highly ionized oxygen atoms, and that's why the structure is polar. Now let's see how a nucleotide structure looks like. To the C1 carbon of the sugar, either deoxyribose or ribose sugar, depending upon whether it is RNA or DNA nucleotide, this C1 carbon is attached to N9 of the purine, adenine or guanine, via glycosidic bond. And on the other hand, via carbon number 5, it is attached to the phosphate group through the ester bond. And in the case of pyrimidines, C1 carbon of the sugar is attached to N1 of the pyrimidine, cytosine, thymine, or uracil, and 5 carbon, it is attached to the phosphate group via ester bond. Now, if you attach adenine plus sugar, which can be ribose or deoxyribose. So if it is a ribose sugar, this will be adenosine. Attach 1 phosphate to it, it will become adenosine monophosphate. Attach 2 phosphates and it becomes adenosine diphosphate. Attach 3 phosphates and it becomes adenosine triphosphate. Similarly, we have ATP, CTP, GTP, and UTP. and these are called NTPs for RNA. For DNA, we have the sugar is deoxyribose sugar. And this is the only difference. And the nucleotides there are called DNTPs. So we call them DATP, DCTP, DGTP, and DTTP. The only difference between the nucleotides of RNA and DNA are the 2' sugar, the 2' position. Sugar in case of RNA is ribose sugar, in case of DNA is deoxyribose sugar. And that's all for the video. Thanks for watching.